It's my pleasure today to present my research results on detection and antibiotic susceptibility of Vibrio cholera in Oreochromis tanganyikae and water in Lake Tanganyika, Kigoma, Tanzania. My name is Martin Michael from Sokoine University of Agriculture in Morogoro, Tanzania. By introduction, Vibrio cholera, this is the gram-negative bacteria that cause severe and lethal human diarrhea disease called cholera. Vibrio cholera can be classified according to phenotypic and genotypic fingerprinting. Phenotypically, Vibrio cholera can be classified by biochemical test, serology, biotyping, phenotyping, and antimicrobial susceptibility testing. In biotyping, Vibrio cholera, in serological tests, Vibrio cholera are divided into 3 serotypes, that is O1, O139, and non-O1 and non-O139. These 3 serotypes of Vibrio cholera, or serogroups of Vibrio cholera, they are very, very toxigenic and very harmful, and they are based in African countries, especially sub-Saharan countries. But also, we deal with O1, Vibrio cholera O1, because it's the one which was commonly reported in East African countries, especially Tanzania, Kenya, Uganda, and Rwanda. And O1 are divided into Ogawa, Inaba, and Hikojima, but in my case, I deal with Ogawa, which produce antigen A and B, subunit antigen A and B, which are very, very toxic antigen to human being. But also, Vibrio cholera has 12 species and 2 serotypes, O1 and O139, which are very harmful to human beings. And the Vibrio cholera in their genome, they have 4 million KBs. They have 4 million genes which are responsible for virulence and evolution of Vibrio cholera, but also they are responsible in antimicrobial resistance. And it has been divided into chromosome number 1 and chromosome number 2. The chromosome number 1 is the one which is responsible for antimicrobial resistance and virulence characteristics of Vibrio cholera. But chromosome number 2 is the one which is responsible for the evolution of Vibrio cholera because it has integron Iceland, which has ability to accept the mobile genetic elements from the environment so that the Vibrio cholera can adapt to their environment. But also, cholera is considered to be endemic in more than 60 countries based in Caribbean, Asia, and African countries. It is estimated that there are about 3 million cholera cases per year worldwide. In 2016, Tanzania experienced the outbreak in Mwanza, Kigoma, Tanzania, and fish were considered to be crucial role in transmission of cholera pathogen. WHO in 2017 suggested that warm water is likely the important habitat for L-tor variant of Vibrio cholera O1. The objective of my study is to evaluate the magnitude of Vibrio cholera and the antibiotic susceptibility patterns in Tanganyika tilapia or Oreochromis tanganyikae and water in Lake Tanganyika during non-outbreak period. I used the cross-sectional study whereby data were collected in 2 seasons, rainy season and dry season. And a total of 140 fish sample and 60 water sample were collected. A culture, biochemical test, serology, and molecular-based method whereby I used the PCR, multiplex PCR, targeting 2 genes, outer membrane protein, the gene coding for outer membrane protein as a species-specific gene, and CTXA gene, the gene coding for cholera toxin gene, as a marker to identify if the Vibrio cholera is toxigenic or non-toxigenic. But also I used the disk diffusion method as a phenotypic fingerprinting for antimicrobial susceptibility pattern. I found that overall prevalence of Vibrio cholera in tilapia was 27.9%, while in water was 33.3%. And in dry season, I found that the Vibrio cholera in tilapia was 18.57, and in water was 26.66. But in rain season, the Vibrio cholera prevalence of Vibrio cholera was 37.14, and in water was 40. But also the proportion of Vibrio cholera in fish gills was 10.04%, while in intestine was 0.8. And their difference is significant at p-value less than 0.01 And the difference between dry season and wet season is significant at a prevalence of 0.018. And this is the graph, or is the figure showing the proportion of toxigenic and non-toxigenic Vibrio cholera. As you see there, the toxigenic Vibrio cholera, they are very, very sensitive, and it is not easier to get them at a non-epidemic period. But also, as you see here, antibiotic susceptibility, gentamicin, ciprofloxacin, and cefotaxime, they performed well as compared to amoxicillin, streptomycin, and ampicillin. This shows that Vibrio cholera, they were resistant to streptomycin, amoxicillin, ampicillin, and the trimethoprim. In discussion, the proportion of fish samples infected with Vibrio cholera in rain season was high compared to dry season. This implies that rain season have ability to increase the chances of contamination and colonization of Vibrio cholera. But the prevalence of Vibrio cholera in fish, gills, and water was high compared to intestine, because in the fish intestine there are accessory toxins which make Vibrio cholera not to propagate well in fish intestine as compared to gills. But also there is high competition in fish intestine as compared to gills. Also, resistance to antibiotic may be related to the abuse, environmental pollution, and overuse in human and veterinary medicine. Study revealed that tilapia and water in Lake Tanganyika harbor toxigenic drug resistance, Vibrio cholera of epidemic potential. And this study recommends the further scientific studies using next-generation sequencing platform to establish the genotypic factors influencing pathogenicity, endemicity, and antimicrobial resistance of Vibrio cholera. Thank you very much.